Okay, now we can begin our discussion by considering specimen collection, reception, recording, and storage. So we can begin. We see what is involved under specimen collection. Okay, so now this is the abstract. The same thing that I'm going to explain is what happens during laboratory work. There are 4 steps that we can consider when we want to obtain a very good quality specimen for making a diagnosis in the laboratory. So these are the main 4 steps that we should consider as laboratory workers. The first thing is make sure the patient is prepared. Okay, the preparation of a patient is very much important. Okay, then after the patient has prepared, then the collection of specimen. When the specimen is collected, it's being processed. When being processed, then storage or transporting the specimen. So there are some principle or a principle of correcting a specimen. So this is the principle. The main key why the specimen has to be corrected in a correct way. The appropriate should be appropriate to the patient's clinical presentation. Okay, so we need to make sure that we instruct the patient to give us the correct sample. For example, let's say if the doctor has requested for blood culture, we can't collect urine, we have to correct blood sample, not urine. So we need to collect the appropriate specimen for the clinical presentation. Let's say we want to do the blood counting. Blood counting, we know that we have to collect the blood sample in a paper top container. So since it's blood sample, so can we use a red top container? No, we can't use, because though a red top container, it can be used to collect blood, but it can be a clotted blood which cannot be of use when it comes to full blood count. So we make sure we collect a correct specimen according to the clinical presentation that the person is presenting. We need to collect at the right time. Okay, there are some specimens that does not require collecting anyhow or at any time. It requires a specific time allocated to that specimen. For example, when we want the sputum for TB, what is the appropriate time? It's early in the morning, just after waking up from the bed. That's when the person can collect the sputum. Then urine, also it needs the early urine. That's what it means. It has to be collected at the right time, not any time. No. Then the next point is collected in a way that minimize contamination. For example, let's say we want to do the stool culturing. Want to make sure that when collecting, the environment where the sample is being collected, it has to be disinfected thoroughly, because if not, we can find that instead of identifying the organism that is causing the problem in the patient, we may end up identifying any other organism that were collected from any other source. The organism that can be identified, maybe it was just a contaminant. So we need to make sure that we collect in a way that minimizes the contamination. Also, we should use the collect equipment. Okay, so when collecting samples, we have different types of containers, and each container is appropriate for the specific sample. Just like I said, we use different containers for collecting blood sample for a specific test. We can't use a red top container when we want to collect the blood sample for ESR or full blood count or malaria test. So we make sure that we know the types of containers that we should use, of which I'm going to show you in this lecture. Documented clearly using appropriate forms. Okay, so there are some forms that each hospital has where the details of a patient are being written. Okay, so make sure that the name is written, the age, and the sex has to be written on that form. The condition, the type of clinical presentation, it has to be written. So make sure that all the details of a patient is being written. That's on the correct form. So now, when transporting the specimen, the transportation is very vital, and how specimens are being stored. For example, the urogenital specimen, it has to be stored in a certain preservative in order to preserve the organism that has to be identified. So we should pay attention to how we transport, because if we are transporting the specimen carelessly, what can be the result? We may end up affecting the public. So those are the things that we should pay attention. We may end up affecting the public or killing the organism of interest if it is not properly stored. That means the temperature has to be taken into place, and preservatives. Then specimen should be collected before medication. So this should be given to the patient. Okay, if a patient has taken medicine and then the sample wants to be corrected, what can happen? That medicine can hide the organism such that at the time of correcting the sample, we may end up of having a false result. So make sure when correcting the sample, no medication is being taken. Okay, so this is how it should be done. Now let's see different types. So these are the main samples that are collected and being processed. These are the main samples. For example, we have sputum, okay, we have stool, blood, urine, pus, urogenital specimens, and body fluid. ear and the eye, nose and throat swab specimens. These are the common specimens that we can collect in the laboratory. So now let's see the requirements of containers and other that we need to use when we are dealing with specimen collection. Of course, we need the urine container. This is where urine is being collected. We need a red top container, a purple top container, a gray top container, green top container, yellow top container. These are very much important for the specific, and I will explain why these has to be appropriate. We have a stool container, sputum container. We need to have methylated spirit for disinfecting the environment and the place where you're collecting the sample, for example, the blood sample. We need to have a cotton wool and a sterile swab. Okay, a sterile swab. We need to have a speculum. Okay, a speculum should need to have it. We need to have syringes. We should have tourniquets. We need to have the tourniquets, syringes, biohazard bag, and also a transporting media. This is where we transport the specimen. When we collect, we put in a transporting media to preserve the organism's life so that it can be identified. So now let's see the methods that we can use when collecting the specimen. Okay, the methods that we can use. For example, we have urine. So how do we collect? Of course, we need to use a sterile container. Sterile means a container free from contamination. Okay, free from contamination, it has to be used. And then, like I said, we need to collect a midstream urine, meaning the early morning urine. This is the type of urine that we have to collect. Then we have to collect using aspartic techniques to collect the urine. Aspartic means we need to make sure that the patient is not contaminating the urine. That's what aspartic means. We go to stool. How stool can be collected? We also use a clean container of sufficient size with a very tight lid, with a leak-proof lid. It should be enough and very tight, because stool smells badly. Okay, then a sterile swab can be used in case of rectal swab. In case of rectal swab, when mostly used when we are identifying certain parasites. Let's go to cerebrospinal fluid. Cerebrospinal fluid. This is the fluid from the spinal cord. What do we use? We use lumbar puncture or spinal tap. Cerebrospinal fluid, spinal tap or lumbar puncture. That's the method we use to collect. So when collecting the cerebrospinal fluid, we use 3 containers, meaning we use 3 containers, meaning we need to collect enough, a little bit of enough, because collecting the sample from the spinal cord is very painful. So when we collect the sample, it has to be divided into 3 plain tubes, because it requires 3 types of tests going to different labs. Okay, 2 for microbiological tests and one for biological analysis, biochemical analysis, and two to the microbiology lab. When we want to collect a swab, it can be an eye, the nose, ear, or throat swab sample. What do we need to use? We use the sterile swab. When collecting, okay, then after using it, we put in that, of which I'm going to show you guys at the end of this video each type of the container that I have been talking about. Urogenital swabs, okay, it's from the vaginal swab, okay, it's eye vaginal swab. We use a sterile swab, okay, to collect the eye vaginal swab. We use a sterile swab. Then the endocervical swab, we use also a sterile swab and a speculum. The urethral swab is also a sterile swab. What of collecting the pus? We also use the swabs and the syringes. You know what pus is? Okay, it's just the accumulation of white blood cells and the damaged cell. So now when we are collecting that, we use a very sterile swabs and syringes. When collecting blood sample, we use different types of container, okay. So for example, we use an ADTA, which is a paper top container. This container is mostly used for collecting blood sample for hematological tests, okay. When talk of hematological tests, we are talking of most of the tests that are done under hematology lab, such as sickling test, ESR, and more, such as also solubility test. Also used for blood grouping, which is under hematology, which is blood bank. Also for viral loads and CD4 test count, EDTA. This is the paper top container. A plain red top, okay, a plain red top container. So what I mean to say a plain red top container, it means there is no additive or chemical inside that container. Unlike the paper top container, it contains EDTA additive, which prevents blood from clotting. But in a red top container, there is nothing in it. It's just empty. Mostly it's used for cross matching and serological testing, okay, cross matching and blood bank and serological testing. We can have also lithium heparin. Lithium heparin, this is the container that is mostly used for biological biochemical testings, okay. These are biochemical testing. It has this same additive, which is lithium heparin. It is an additive. And then we need to use a gray top container, which is specifically for glucose testing, okay. It is specified for glucose testing. So when we use the appropriate container, the appropriate method, and give the correct instruction to the patient, we can collect a very good sample for the patient, okay. So when we collect those samples, what do we need to do? First, we need to write the name of a patient, okay? The name of a patient on the sample. We write the age, the sex, the words, specimen type. Also need to write the date and the time at which that specimen has been collected. Then when it comes to storage, urine, it has to be stored at 40 degrees Celsius. This is the temperature where it has to be stored. Then stool, there must be a transport media where stool is being put. For example, rectal swab, it has a transport media in the same swab where it is being transported. Okay. And stool, it shouldn't be stored for more than 24 hours. It can only stay for a day. Then cerebrospinal fluid, which is CSF, it has to be kept at room temperature. What should be the room temperature? It's between 20 degrees to 25. So that's where it should be kept. And blood should be kept between 2 degrees to -8 degrees Celsius, any blood sample. Then urogenital specimen, it has to be stored at a temperature of 2 degrees Celsius to 30 degrees Celsius within 14 days of collection. Alternatively, okay, specimen may be stored at -20 degrees Celsius. But for the eye vaginal fluid, because the organisms that are identified in eye vaginal fluid is very sensitive. It can die within minutes. So as soon as it is being collected, it has to be transported immediately to the lab. So make sure that the sample that is being collected is sufficient enough, and we should always try to avoid working with sample that has been overstayed, or a sample that is clotted. If the sample shouldn't need to clot, we should make sure that we deal with a very good sample, and then it should, for us to get the result that we need from the patient. So in the next video, we are going to do the blood bank. We are going to see the ABO and RH forward blood grouping. So we are going to see how we can group blood.